Welcome to the lecture series in food biotechnology. This is the third part in the series where we are dealing with food fermentation, its nutritional benefits as well as the health benefits. See, fermentation is something which naturally happens, and what we call as a food is a combination of carbohydrate, starch, protein. So, all of them can be contaminated by microbes, and remember, microbes are prevalent all throughout atmosphere. Wherever we are talking about, the microbes are present there. So, all food raw materials can be contaminated by microorganisms, and when in general these microbial reactions result in food spoilage, but in many cases microbial fermentation can lead to specific taste and texture in food. See, for the microbes, the food is actually an extremely nutritive substrate. So, when the microbe happens to fall on the food material, it will utilize the nutrition in that and start multiplying. So, as it multiplies, its metabolic activity is going on, the food substance gets broken down, and this broken down material can be absorbed by the organism for its life, can be converted into something else, and then of course the microbial waste products also get excreted. Now, depending upon what is the sort of the waste product which is there, it can be a food spoilage, when especially when we talk about production of hydrogen sulfide, proteins getting decaying, putrefaction, etc. There we are talking about the spoilage, because such food is not palatable by humans. The second aspect is the presence of certain metabolites can cause food poisoning, but in many cases when these microbes grow, their metabolic products can add up flavor, especially if it is a fermentation process. It can add up flavor or give a good aroma which will give a specific taste and texture to the food. So, in most of the cases microbial fermentation can lead to specific taste, specific texture, and the final product of fermentation is the result of unique chemical, physical and the biological interactions between specific microorganisms and the food. So, finally, what is the flavor which is residual, which is present in the food, or what is the texture which is present, depends upon the organism. The moment we call microbes, that food can be utilized by n number of microbes which are present. Each microbe can give its unique texture, its unique flavor. So, when we have to have a marketing a product saying this food has this texture or has this unique flavor or has this particular taste, for that you require a specific microbe. So, the nature of the food as well as the interaction of the microbe with that particular food will give that specific taste or texture. Earlier, fermentation was seen as a generic process to preserve raw materials and to transform its sensory characteristics, but now these days it is intentionally used to add finer functional assets such as increased nutritional, health, psychological and probiotic benefits, as well as add regional nuances and individuality with respect to its sensory character. See, earlier we did not have much of the preservation techniques, and most of the food material are seasonal. For example, when we talk about harvesting fruits, harvesting vegetables or harvesting cereals, it is seasonal. So, to store it from one season to the next season and to utilize it during the time when it is not growing, we used to ferment, so that makes it easy to store. But nowadays, rather than store, we have so many processes where we can, so many phenomena have been discovered, so many how we can preserve the food, so much of technical knowledge has come. But now the fermentation, in addition to storage, is also used to increase the nutritional aspect, to give health benefits. In some cases, in many cases, whatever is the raw material which is present in the food, once it is broken down by the microbes, would be better available for the humans, that is what we call as the bioavailability, that increases when there is a microbial fermentation there. So, and also in each part of the world they have a specific food, we call it as a regional preference. So, that specific food, that particular taste comes because of a specific microbe which they use for the fermentation purpose. So, for this we need to know the microbial ecology of fermentation. So, the basic questions which we ask here are, what are the species present? So, if this is the food, for example, my food contains carbohydrate and protein, that is a major portion. So, if this is the food, then in this food which are the microorganisms which will live, which can use this? What are the species which are present? Not only the genus but also the species which are present. Now, among the species or among the microbiota which are present, are all of these essential? Remember, we are talking about a product. So, for this product to make it palatable to the consumer, do we require this many amount of the genera to be present? Are all of them essential? The next question to be asked is, do some of these genera which is present, are they detrimental? Meaning, when they grow, do they give some metabolic byproduct which is not required, or does their growth inhibit the bioavailability of the product, or does it give a wrong flavor or a not so acceptable aroma or a texture? So, what is it is detrimental? Where do these microorganisms come from? Is it specific or is it like, and also what is the sort of modification which happens? So, does the raw material, the food, does it itself has the microbe, or are we adding the microbe during food processing? Are we adding a pure culture or is it a mixture? So, all those questions come there. And then finally, what are the factors which control the survival of these microorganisms and their growth? See, when we do the processing, for example, high temperature, or we add so many substances which can change the pH. So, all these preservation processing techniques which we use, how do they affect the microbiota which is present in the food? Will they survive in these modified processing techniques? Will they exist? What sort of a modification? Does it change their metabolism? Because if they change their metabolism, then it means that the flavor or the texture will also change along with that. So, all these are the questions which a food biotechnologist has to look for, especially if he is going to have a fermented food product. So, also relevant is the knowledge of the activity, that is the microbial activity, and the impact of the metabolic activity on production and product quality. See, for example, when a microbe ferments, the initial fermentation product and after complete fermentation what happens to the food, they would be completely different. So, this depends upon not only the quantity of the substrate which is present, but also on the quantity of the culture microbiota which is present and how many hours of fermentation has happened. Are you allowing partial fermentation, or are you stopping in between, or are you going to make the food product after complete fermentation? So, for that we need to know the metabolic activity, what are the byproducts and what happens towards the end. And for most of the fermented products, the discovery of the microbial ecology is actually very challenging, as the microbial communities which is present in each food is usually very diverse. The physical, chemical and the biological properties of the food matrix is also very complex. See, when you have a community of microbiota which live on a food substrate, each of these microbes will interact with the food, and because the other microbiota is present, the metabolic activity of one genus or one species will also interact with the other organisms which are present, and along with it each of this will cause changes to the food. So, this is actually a very complex interaction which has to be studied very well to know the final product, to know when you design it. The most common microorganisms which are involved in food fermentation are bacteria, yeast and molds. Among bacteria, Lactobacilliaceae and the Azotobacter species are the ones which are normally seen. Among fungi, we have yeast, and yeast like fungi, for example Saccharomyces cerevisiae, which very commonly we use for making bread, molds like Penicillium species, which are associated with the ripening and the flavoring of cheese. Now, what is the role of fermentation in food processing? One, we already saw that it was for preservation. The second aspect is fermentation enriches the human diet, because see, when we acquire flavor and texture is something which is acquired. So, each food as it undergoes fermentation, this microbial byproduct and the association of the microbe with the food can change its flavor, its texture, its aroma, which would make the food more palatable, and maybe for in some consumers they would appreciate the food more if the fermentation level and the texture is much more. A typical example we talk about is cocoa fermentation. This is something which all people love, chocolates. It's the major raw substrate is actually the fruit of cocoa tree. So here, when this cocoa, the raw cocoa which you get from the tree undergoes fermentation, different levels of fermentation can give rise to different products. Ultimately, the chocolates would be of like, it can be starting from your normal chocolate to the caramel, to the bitter chocolate, to the dark chocolate. So each one, that specific flavor and the texture is because of the different levels of fermentation which is normally seen. For example, the sugars in the cocoa, if they gets fermented by yeast, they give rise to carbon dioxide and water. Or if it is too much of fermentation, it will give rise to ethanol and carbon dioxide. Now, these sugars can be acted upon by both lactic acid bacteria as well as acetic acid bacteria. So, if lactic acid bacteria act upon the sugars, your product is going to be lactic acid, which is non-volatile. Okay? If it is the acetic acid bacteria, again you get acetic acid, which is volatile. So, it gives an aroma. Now, this actually develops the product, end product of fermentation here will have a bitter astringent taste here with a grayish color. Now, this taste which will develop a flavor precursor, because again further fermentation breaks down the amino acids and it frees up the amino acids and frees up the peptides, which gives a what you call as the dark chocolate or even the bitter chocolate or the specific levels. So, different levels of fermentation actually can give rise to different types of products with specific texture, flavor, aroma. So, when we talk about preservation of food, the different techniques which are used are lactic acid fermentations, alcoholic fermentation, acetic acid fermentations, alkaline fermentations, and fermentation using in very high salt concentration, where we use 13% of salt concentrations. Typical example is your pickles, where we can pick and fermented vegetables like sauerkraut, etc. Dill, what you call as pickled cucumber. So, all these are preserved food where fermentation is the process which is used. So, here we enrich the food substrates biologically with vitamins, essential amino acids and essential fatty acids, because when the microbes break down the food product, the byproducts can be vitamins, essential amino acids, which would be now freely more bioavailable to the humans who consume them. Then, if there are any toxins which are present, fermentation process can detoxify those toxins which are present, and again fermented products are easy to cook. So, a decrease in the cooking time as well as a decrease in the fuel which is required, that is also an added advantage of food fermentation. So, now let us see what are the different classifications of food fermentation. So, different types of classification occurs in the case of food. One is by the category, as we saw earlier, the alcoholic beverages, the alcoholic fermentation, which is fermented, the organism used mostly are the yeast. Then you have the vinegars. Here the organism is Acetobacter. Milk, yogurt, cheese, yogurt, for all this we require fermentation by lactobacilli. Fish and meat, again the fermentation is by lactobacilli. Plant proteins, these plant proteins are usually fermented either by the moles along with or without lactobacilli and yeast. So, plant products can have two types of fermentation. It can be just fermentation with moles alone, or you can add a lactobacillus to the mixture and yeast to the mixture. So each of this, depending upon the combination which you use, will give you different types of fermentation byproducts. So by classes, so beverage, cereal products, dairy products, fish products, fruit and vegetable products, legumes, meat products. By commodity, so we talk about fermented starchy roots, fermented cereals, alcoholic beverages, fermented vegetable proteins, fermented animal proteins. And finally, what most of the people follow is the functional basis of the fermented food, the staples which we consume every day, breakfast, so porridges, bread, etc. Then the sauces and the relishes which we use as a side dish for the staple for the bread. Then opaque or clear beer, wines, meads and other alcoholic drinks. Then food, fermented food which is used for special occasions. So this actually, the general functional classification, this is a traditional Sudanese classification based on the type of food, what is it used for. Is it a staple one or is it something which you use as a supplement for the food? So this is the normal functional classification of the fermented food. So generally classification is fermentations producing textured vegetable protein, meat substitutes in legume, cereal mixtures. For example, vegans or vegetarians do not consume animal proteins, fish, meat, etc. So for them you require a rich protein source, or meat substitutes can also be used. So for this, actually, to increase the flavor or to give it a meaty flavor to the food, fermentation, especially soybean, and there are other legumes which when fermented give that particular texture and flavor to the food products. The substrates which we use would be a legume cereal mixture, and to this if you add specific microbes, the byproduct of the fermentation will give rise to textured vegetable product which with a meaty flavor. A typical example is the Indonesian tempeh. The other classification is high salt or savory meat flavored or amino acid flavored peptide sauce and paste fermentation. This is usually used as a relish for your staple like your Chinese soy sauce, the Japanese miso, the Malaysian kecap, etc. are typical examples of these. Lactic acid fermentation. This, as we saw earlier, sauerkraut, cucumber, pickles, all of them follow lactic acid fermentation. Lactic acid fermented milk include the yogurts in the Western world, the Russian kefir, Indian dahi, etc. And in the case of fermented cereal products, we have the Indian idli, sourdough breads, dokla, etc. So there also the organism, see it's a cereal legume mixture in case of dokla, idli, etc., which gets fermented by lactobacilli. The basic fermentation pathway is lactic acid fermentation there to give the specific flavor and texture. Typical example of alcoholic fermentation are the grape wines, honey wines, the Japanese sake, etc. In the case of acetic acid and vinegar fermentation, we have the apple cider, wine vinegars, the coconut water vinegar, etc. A byproduct of the alkaline fermentation is the Nigerian dawa and the African iru. And a typical example of leaven bread product is Western yeast and sourdough breads. So depending upon which type of fermentation the substrate undergoes, we have different types of products based on the organism which causes the fermentation in the particular substrate. There are many microbes which can cause actually food poisoning or food poison, so spoilage also, or as we saw, when the microbe spoils the food, certain byproducts can be there which in turn can act as a toxin causing food allergy or even food poisoning. So in such cases, when we specifically introduce the microbe to cause fermentation, we need to know whether the food is going to be safe. For this, if the food has to be rendered safe, then it means that we need to know whether the organism which we are introducing is safe, what are the byproducts, its metabolic pathways. So food fermentation generally involving the production of lactic acid is considered to be safe. So here the organism converts the fermentable sugars into lactic acid. The normal organisms which we use are Lichenostoc mesenteroids, Lactobacillus brevis, Lactobacillus plantarum, Streptococcus thermophilus, etc. This type of lactic acid fermentation is responsible for processing and preserving large quantities of human food and for assuring its safety. Additional factor for the lactic acid fermentation is that the different organisms like the nucleostock or the lactobacillus, they do the fermentation giving rise to different types of flavors, aroma and textures. So depending upon the organism, the same substrate can give rise to different flavored products. Raw unpasteurized milk will rapidly sour because lactic acid bacteria is present there, and that organism will ferment the lactose into lactic acid. So now what happens, the pH of the, because once the lactose is converted into lactic acid has a low pH. So the pH drops and you have an acid present. So now what happens, the pH drop as well as the presence of the acid will prevent other microorganisms and the pathogens from invading the food or the milk. So they will not be able to colonize the milk, in which case your milk is now, even though the milk has soured or it gives a bitter sour taste, other microbes or the spoilage microbes or the poisonous microbes cannot live in them. So the food is now safe. If the whey is allowed to escape or evaporate, the residual curd becomes what we now call as a primitive cheese. A typical example of the lactic acid vegetable fermentation is sauerkraut. Here the substrate is fresh cabbage. So the cabbage is shredded and we add 2.25% salt to the cabbage, and in this the organism first which grows is the leuconostoc mesenteroids. So what will it do? The leuconostoc which grows in this mixture produces lactic acid, acetic acid and carbon dioxide. So the release of this carbon dioxide will remove all oxygen which is present. So now what happens? The fermentation now becomes an anaerobic fermentation. In this scenario, Lactobacillus brevis grows. So leuconostoc has done its metabolic pathway, making the condition there anaerobic. Now Lactobacillus brevis will grow in this anaerobic condition, producing more acid. And then again the last organism which grows is Lactobacillus plantarum. It produces even more lactic acid and the pH now drops to below 4. So under the anaerobic condition and at a pH of 4, any vegetable, I mean it needn't be cabbage, but any vegetable which is preserved in this will stay long for a long time as long as that anaerobic condition is maintained. But the moment it comes, you open up air and the oxygen comes in contact, that anaerobic condition goes off. So then it becomes an easy substrate for other organisms to grow. But as long as this pH and as long as this anaerobic condition is maintained, the vegetables can be preserved for a longer time. Another example is the Korean kimchi. It is similar to sauerkraut, but there the vegetables which we add, the Chinese cabbage, garlic, radish, red pepper and ginger. The fermentation process it is less acidic than sauerkraut. Actually speaking, its consumption is while it is carbonated. Another typical example of lactic acid fermentation are what we call as the pit fermentations. This has been followed for centuries to preserve taro, banana, cassava tubers, etc. So what they do is they make a pit in the ground and the pits are actually lined with leaves, and the fermented paste or the punctured fruit or the tuber is placed in these leaf lined pits. The pit will then be covered by leaves and then it will be sealed. So it is now we are maintaining an anaerobic condition inside the pit, and since it is a fermented paste or the punctured fruit, the organism will ferment it, converting into lactic acid. So as low pH will be there, the automatic result. This will maintain the stability of the food inside as long as we maintain the low pH and as long as we maintain the anaerobic condition. Lactic acid fermented rice, shrimp, fish mixtures. A typical example is the Philippine balao balao. This is a mixture where boiled rice, raw shrimp and salt is packed in an anaerobic condition, and then it will be allowed to ferment for several days. So because of this fermentation, the chitin shell of the shrimp will become softened. So when you cook this, remember we earlier said that fermentation will reduce the cooking time. So because of this, the chitinaceous shell, which otherwise cannot be cooked, because it is now softened, it gets easily cooked, and along with the exoskeleton, which is the chitin, the shrimp can be consumed. The lactic acid bacteria generally produce several antimicrobial compounds such as organic acids, hydrogen peroxide, carbon dioxide, diacetyl, and also broad spectrum antimicrobials such as ruterin and the production of bacteriocins. So, these antimicrobial compounds also give further protection to the food. The antimicrobial effects of the organic acids like the lactic acid, the acetic acid and the propionic acid is believed to result from the action of the acids on the bacterial cytoplasmic membrane, which interferes with the maintenance of the membrane potential and inhibits the active transport. And generally, acetic acid is more inhibitory than lactic acid, and it can inhibit yeast, mold and bacteria. Propionic acid inhibits fungi and also some gram-positive bacteria. The contribution of acetaldehyde and ethanol to biopreservation is actually minor, since the flavor threshold is much lower than the levels which are considered necessary to achieve the active inhibition of microorganisms. Food where you have the presence of ethanol or acetaldehyde, the flavor will be different, and in many cases it will not be palatable or people will not prefer that. And for using or preserving food using these, we require a higher concentration of these chemicals, and if a higher concentration is present, the palatability of the food goes down. Hence, inhibition of microorganisms will not normally work. Hydrogen peroxide is usually generated during lactic acid fermentation, and that can be inhibitory to certain microorganisms. And this inhibition is because of the oxidizing effect on the membrane lipids and cell proteins. See, H2O2 can break down to hydrogen and H2O water, and a free radical oxygen is released, what we call as reactive oxygen species. The presence of reactive oxygen species can go, or it can oxidize the membrane lipids and the cell proteins, changing the oxidation potential. The membrane potential is lost, the cell wall breaks, so thereby destroying the organism. And also this hydrogen peroxide can activate what we call as the lactoperoxidase system of the fresh milk, because of which the fresh milk remains or inhibits microbial activity. Alcoholic fermentations. Fermentations which involve the production of ethanol are generally safe. The products usually include wines and beers. Here the organism which is involved is yeast fermentation, but we can also involve yeast like molds like Amylomyces roxy and mold like yeast like the Entomycopus. The substrates include diluted honey, sugarcane juice, palm sap, fruit juices, germinated cereal grains, hydrolyzed starch, etc. All these products contain fermentable sugars. So, the organisms will convert the fermentable sugars into ethanol by natural fermentation in the and the yeast are present in the environment. Ethanol has a germicidal effect, and as long as the product is kept in anaerobic condition, it will be stable, it will be preserved. With starchy substrates like grains, the starch, some of the starches initially gets converted into fermentable sugar. So, for this usually in many cases in traditional food, the grains are chewed first. So, this produces what we call as a ptyalin. This happens in the Andes region of South America. So, they chew the maize so that it produces ptyalin, and then it gets fermented, or even malting, which we use in barley, or in most of the cases with the grains where we have production of beer from grains. Acetic acid or vinegar fermentation. If the products of the alcoholic fermentation is exposed to air, the acetobacter, the organism which is present in the atmosphere, can oxidize portions of ethanol to acetic acid and vinegar. Fermentation involving the production of acetic acid yields food or condiments which are generally safe, and acetic acid is also bacteriostatic to bactericidal. That is, bacteriostatic meaning it inhibits the multiplication of bacteria. So, as long as a particular concentration of acetic acid is present, the bacterial multiplication would be inhibited. Or bactericidal meaning at higher concentrations it can destroy the organism. This depends upon the concentration of the acetic acid which is present there, and it has more preservative properties than ethanol. So, normally for pickling and preserving cucumbers and vegetables, we use acetic acid rather than ethanol. Leavened bread fermentations. These are closely related to alcoholic fermentations. They are safe, and because of the short fermentation time, the ethanol which is produced is a minor product, but the carbon dioxide which is released actually gives an anaerobic condition here. When you bake it, it gives a surface resistance which will give resistance to the invading organisms from the atmosphere. And baking also destroys the microbes which are present in the bread. And alkaline fermentations are generally safe. Most of the African foods are products of alkaline fermentation. Typical example is the natto, Thai food, etc. We will continue the products and the pathways of metabolic fermentation in the next part. Thank you.